Genetic engineering. First question we're going to answer is, how does genetic engineering work? First, we start with a DNA strand, and with a restriction enzyme such as EcoR1, we can split the DNA strand into the multiple gene components. With these DNA strands, they can be inserted into a bacterial vector, which is split with the same restriction enzyme. It is then attached with DNA ligase that works sort of like tape. The end product is plasmids with the wanted or unwanted gene, or an unchanged plasmid. This is a bacterial cell. Inside the cell, there is a bacterial genome. When we try to insert the plasmid from before into the bacterial cell, it could end up killing the cell. The bacterial vector can be safely inserted if the temperature is first slowly cooled and then rapidly heated, which creates holes inside the bacterial walls, and then the bacterial vector can be safely inserted, not killing the cell. Then overnight the holes will seal up. When a cell grows, it can either have the correct gene, the wrong gene, a plain plasmid, or no plasmid at all, but the latter is prevented by having a special coating on the agar plate which prevents it from growing. The end product is lots of copies of the desired gene. Once split, electrophoresis is used to separate the different genes. They are placed into a buffer, and currents force the genes to move across the gel towards the electrode. The speed based on the gene's weight and size is what sorts it. Applications, and there are many of them, including industrial, agricultural, and many others. First, we will start with agricultural. The applications include salt tolerance, pest resistance, disease resistance, more crop yields, and less water usage. The industrial applications include parental disputes, forensic analysis, medical diagnosis, genome mapping, and gene sequencing. The medical applications include making vaccines, human insulin, interferon, growth hormones, and identifying diseases. Advantages. There are many of them for multiple kinds of people. It can help identify parents, analyze crime scenes, and map/sequence genes. In agriculture, it can help to make more nutrient-rich soil, better nitrogen fixing, disease resistance, less soil damage, and more efficient nutrient usage. It also can help with the production of vaccines, human insulin, interferon, growth hormones, and identify diseases. Disadvantages. Although it can have its benefits, it can also have some devastating side effects. The disadvantages include disturbing the delicate balance of biodiversity, being harmful to an organism higher up in the food chain, eating GMOs may be harmful to human health and cause allergies, it might replace traditional farming and meat production practices, the misuse of this technology may result in the production of biological weapons. It could make genetically modified humans, which may interfere with the diversity of the human genetic pool. And finally, the ethical and moral issues with some religions on this technology. Thanks for watching, guys. Credits go to MIT, K-12 Videos, Life Noggin, and Vsauce.